All right, we're about to jump into a really cool scientific detective story. We're on the hunt for suspects that we can't see, we can't touch, and we definitely can't get close to. So the big question is, how do you do that?
I mean, really think about it. You're an astronomer, right? And you're looking at a star that's millions of light years away, and you want to know what it's made of. You can't exactly send a probe out there, you can't grab a sample, so how in the world do you solve that mystery? Well, that's the huge challenge we're going to unpack today.
The secret, it turns out, is in a kind of cosmic forensics. Every single molecule out there, from something simple like water to a really complex organic compound, has its own unique signature. We call it a molecular fingerprint, and it leaves this fingerprint on the light that it interacts with. Okay, so first part of our investigation. We need to figure out how to actually read these cosmic clues. This is where we get into the fundamental techniques of our detective work.
Our main tool is called rotational spectroscopy. Now, that might sound super complicated, but the basic idea is actually pretty straightforward. See, as molecules are tumbling and spinning around in space, they absorb light, but only at very, very specific frequencies. So when we look at that light, we see a pattern of dark lines where that energy got soaked up. It honestly looks a lot like a barcode. And that unique barcode, that is the molecular fingerprint. Now, in a perfect world, in the textbook version of this, we'd be working with what paper 01 calls the rigid rotor model. This is where we just assume the molecule is a perfect, solid little object, like a tiny dumbbell that never changes shape. And in that ideal case, its fingerprint is super clean with these nice, evenly spaced lines that are incredibly easy to read. But, as you can probably guess, reality is rarely that simple.
And just like any good detective knows, real-world evidence is almost never clean. The clues get messy, and our nice, perfect fingerprints, they can get seriously smudged.
So here's problem number one. Real molecules aren't perfectly rigid. Not at all. As they spin faster and faster, centrifugal force actually makes their bonds stretch out. And according to paper 01, this little stretching effect squishes the spectral lines closer together, completely distorting that neat pattern we were hoping for. All of a sudden, our crystal clear fingerprint is pretty smudged. And the bond stretching? That's not the only problem. The clues get even messier when we start to think about another really important factor, the molecule's actual three-dimensional shape. You see, not all molecules are simple straight lines like a pencil. Research from paper 05 points out that more complex shapes, which they call symmetric tops, just picture a spinning toy top, they tumble in much more complicated ways. And this creates a way more intricate overlapping pattern of spectral lines, which makes that fingerprint incredibly difficult to figure out. So, we've got smudged prints and super complex patterns to deal with. But now we get to the most dramatic twist in our entire investigation. This is a discovery that really calls the whole foundation of our detective work into question. The entire method we've been talking about relies on this one huge assumption, that one fingerprint belongs to one and only one molecule. But what if that's not actually true? Researchers in paper 04 started asking that exact question. Does a rotational spectrum really uniquely identify a molecule every single time?
The answer sent a shockwave through the scientific community. It turns out, the answer is not always. And that one little discovery throws a giant wrench into decades of this kind of forensic work.
This is what paper 04 calls the twin problem, and it's a big deal. Scientists found out that completely different molecules can, just by total coincidence, produce rotational spectra that are nearly identical. This creates a massive problem of ambiguity. It's basically the molecular equivalent of a forensic nightmare. You've got two different suspects, but they have the exact same fingerprint. Who do you charge?
So with smudged prints, complex patterns, and now identical twins, it kind of seems like our case has gone cold. But this is where modern science gets to call in a brand new partner, a high-tech detective that can solve the cases that leave us humans totally stumped.
And that partner is, you guessed it, artificial intelligence. Specifically, a method laid out in paper 02 called probabilistic deep learning. This is the cutting-edge tool that can see right through all the noise and all the confusion.
So how does this AI detective actually work? Well, it's pretty awesome. We feed it the messiest, most ambiguous spectrum we can find, twins and all. The deep learning model then just sifts through all that data, and it finds these tiny, subtle patterns that are completely invisible to the human eye. And here's the best part. It doesn't just spit out the most likely identity of the molecule, it also gives us a probability score telling us exactly how confident it is that it's got the right guy. The potential here is just enormous. The researchers behind this text say that using AI for this kind of detective work can greatly accelerate the rate of new molecular discoveries. I mean, this could let us find new molecules in the atmospheres of distant planets or in crazy chemical reactions right here on Earth, and do it way faster than we ever thought possible.
Which kind of brings us to our final thought. For decades, we've been trying our best to read these molecular fingerprints, struggling with blurry clues and cases of mistaken identity. But now, with an AI partner that can see with basically superhuman clarity, the whole field is just wide open. So you have to wonder, what are the next great cosmic mysteries we're going to solve?